Hello everyone and welcome to ASA level biology classes. Today it is lecture 3 from chapter 1 cell structure, and in this lecture we are going to learn about the ultrastructure of an animal cell, and we are going to deal with two cell organelles, that is nucleus and endoplasmic reticulum. Let's start with the ultrastructure of the animal cell. Now if this is the picture of an animal cell, and these are the cell organelles which you can see inside the animal cell. Now if you see here, the animal cell has various cell organelles which and plant cells also have a similar kind of cell organelles, but there are certain cell organelles which are exclusively present only in plant cells and not in animal cells. That we will be dealing later on. Today we will be talking about the animal cells and the structures involved with it. We will be first talking about the first structure, that is nucleus. This is the nucleus, what you see here. This is the nucleus, and this nucleus has nucleolus. This is the nucleolus, and what you see over here, these thin thread like structures are called chromatin, and you have there is a nuclear membrane, which is actually double membrane, which is also called as nuclear envelope, and on this nuclear envelope there are nuclear pores. A pore, that means which allow certain materials to go inside and outside. We will be talking about this nucleus in great detail in the next slide. The next structure which we will be talking about is rough endoplasmic reticulum. The rough endoplasmic reticulum, basically endoplasmic reticulum is of two types. One is the rough and the other one is a smooth, as you can see here. Rough endoplasmic reticulum is the organelle which is in continuous with the nucleus, and it has a structure like this. It has a tubular membranal structures like this. It is called as rough because it has ribosomes on it, and why it has ribosomes on it and what is the function, that will be talking later on. The smooth endoplasmic reticulum is smooth because it doesn't have any kind of ribosomes attached to the endoplasmic reticulum. So this is the smooth endoplasmic reticulum, what you see here, and this is the rough endoplasmic reticulum. This is the nucleus. The next cell organelle that we will be talking about is ribosomes. Now ribosomes, as I already told you, it can be freely available in the cytoplasm. As you can see, these dots here, what you see, these dots are nothing but the ribosomes. They are just freely available, and certain ribosomes are attached with the membrane, endoplasmic reticulum membrane, so it is called as not free ribosomes but attached ribosomes. The next organelle that we have it here is mitochondria. This is mitochondria, and this is a very important cell organelle, basically to provide energy, and that energy is known as ATP, which is produced in mitochondria. Again, we will be talking in great detail about this mitochondria in our future classes. The next structure which is found in animal cell is lysosomes. Now lysosomes are also called as suicidal bags. They have lytic enzymes and acid, which is basically helping the cell to digest and engulf the bacteria and worn out cells also. Again, we will be talking about lysosomes later on. Then we have the next organelle which we have it here in animal cell is Golgi bodies. This is this entire structure which you see here, this structure here, these membranal stack like structures are nothing but Golgi body or a Golgi apparatus, and these Golgi apparatus, they keep on producing vesicles, releasing vesicles in the cytoplasm. Then there is So all these structures which we have just now discussed is present in plants as well. And of course there is a plasma membrane, which is basically this membrane which you see all above, all onto the cell. This is a cell surface membrane on the plasma membrane, which is actually selective, which is semi-permeable membrane. And again we will discuss about this membrane later on. All these structures, that is nucleus, endoplasmic reticulum, rough and smooth, mitochondria, lysosomes, Golgi bodies, Golgi vesicle, till now whatever we have discussed is present in plant cells as well. But there is one thing which is not present in plant cell, that is centrosomes. And this centrosome has a centrioles which are lying perpendicular to each other, at right angle to each other, like this. So there is one centriole like this, and there will be one centriole like this. So these centrioles are present, and they generally divide, they just go away from each other, and they basically help during the cell division. And we will be talking about this in greater detail when we will be talking about microtubules and microtubule organizing centers in my future lectures. Now one thing which is very important to understand is that these centrioles which is present in the centrosomes produces microtubules. Okay, and these microtubules are which is actually spindle fibers helping these animal cells to divide. During the mitosis, the chromosomes segregate and they they get divided into two cells because of the presence of these microtubules, and which is produced by these centrosomes. So this was the entire ultrastructure of the animal cell. And what we have observed in this is that apart from chloroplast, animal cells have almost all the cell organelles, and of course cell wall. So cell wall, I'm just going to write down here, cell wall and chloroplast and large vacuoles. Animal cells do have vacuoles, but they are tiny. They are not as bigger as compared to plant cells. Plant cells have very large vacuoles. I'm just writing large vacuoles. So these are the things which are exclusively present in plant cells. So these are exclusively present in plant cell. Okay, so but apart from all these things, everything is present in plant cells as well, but centrioles are not present in plant cells. So I'll just write down here so you will remember it. Centrioles are not present in plant cells. So this is something which is not present in plant cells. Apart from that, all the other organelles what you saw here in animal cells are present in plant cells. Apart from these organelles, plants do have cell wall, chloroplast and large vacuoles. Animals also have vacuoles, but animal cells also have vacuoles, but these vacuoles are not large as large as compared to plant cells. And we will be talking about why do they have large vacuoles, why plants possess large vacuoles. We will talk about this later on. It has a very great significance in plant cells. Now let's take up the next concept, that is we will understand the next cell organelle, that is nucleus. Now nucleus, as I told you, this is you can say that's the brain of the cell, because here only the entire genetic material is present. So it is present inside the cytoplasm, that is where it is located, and I have shown you the location in the animal cell as well. It is the largest organelle in the cell. This is something very important and interesting. Contains thin fibers of DNA and protein called chromatin. So why DNA and protein? Because DNA and histone proteins together, when they coil together in a supercoiled fashion, they form chromatins, And these chromatins, what you see here, these thread-like structures, what you see here is nothing but the chromatin, which is actually a condensed form of DNA, and this condensation happens because of the very special protein called as histone proteins. The next thing is, it is surrounded by two membranes. As I told you, the nucleus is surrounded by two membranes known as nuclear envelope, and you can see this nuclear envelope right here. The outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum, as I have shown you in the previous slide. The nuclear envelope has many small pores which are called as nuclear pores, So what you see over here, these points, these holes here, these are nothing but nuclear pores which are present on the nuclear envelope. And the function of these nuclear pores is that they allow and control the exchange between the nucleus and the cytoplasm. The substances leaving the nucleus, that is mRNA and ribosomes for the protein synthesis. So very important, you need to remember this. The substances which are leaving nucleus, that means which are going outside nucleus, is the substances which are going outside nucleus is mRNA, and we will be talking about mRNA in my future lectures. Why do we need mRNA for protein synthesis, when we will be talking about the central dogma, and ribosomes. So these are the two things which are leaving the nucleus for protein synthesis. Second thing is, the substance which are entering inside the nucleus through nuclear pore is proteins to help make the ribosomes, nucleotides, ATP, and hormones such as thyroid hormone, T3 hormone. So these are the things which are entering inside the nucleus. So things which are entering inside the nucleus, I'm just going to write down here, that is proteins which are helping to make ribosomes, because ribosomes is produced inside the nucleus. So proteins which are making ribosomes, that is the first thing. Second, nucleotides, and third is ATP, adenosine triphosphate, that is the energy currency, because every whatever reactions, whatever things are going inside the cell, whether it's nucleus or any cell organelle, it requires energy. So that is ATP. And fourth is hormone T3. So all these things are going inside the nucleus through nuclear pore. That was about nucleus. And the nucleolus, as I told you, the ribosomes are produced inside the nucleus. So it's a nucleolus which is which is over here, which you see over here, this condensed part, which is the dark part, the dark part which you see over here is nucleolus, and this nucleolus is responsible for making ribosomes, and this ribosome is going out from the nucleus via nuclear pore for the protein synthesis. So the nucleolus makes ribosomes using the information of its own DNA. That is very, very important. So that was about nucleus. The next important cell organelle which we'll be talking about is endoplasmic reticulum. Now, as I told you, endoplasmic reticulum is in continuous with the outer membrane of the nucleus. As you can see here, this is a nucleus which just now we have discussed, and the outer membrane of the nucleus along with it, it is in continuous with the outer membrane of the nucleus. So it's a very complex network of transport channels, and we have two types of endoplasmic reticulum. The first one is smooth endoplasmic reticulum. It is smooth because it doesn't have any ribosomes in it. So this because and it has a very different function. It makes lipids and steroids such as cholesterol and the reproductive hormone estrogen and testosterone. So I'll just highlight here. So ribosomes is free from smooth endoplasmic reticulum, hence it is called as smooth endoplasmic reticulum. It has a different function. It doesn't form protein, but it makes lipid and steroids. Whereas if you see the rough endoplasmic reticulum, it is involved in protein synthesis, but smooth endoplasmic reticulum is involved in lipid and steroid production, such as cholesterol, reproductive hormones like estrogen and testosterone. So this is a basic function of smooth endoplasmic reticulum. The next is rough endoplasmic reticulum. Rough endoplasmic reticulum contains ribosomes and it releases newly made protein. So it releases newly made protein from the cell. So proteins made by the ribosomes on the rough endoplasmic reticulum enter the sac and move through them. The proteins are often modified in some ways on their journey. So small sacs called as vesicles can break off from the ER and these can join with the Golgi bodies. So every time whenever the protein is formed inside these stacks, inside this reticulum and rough endoplasmic reticulum, these proteins will move through these stacks and they come out from these stacks, from these membrane via vesicles, and these vesicles which are coming out from the rough endoplasmic reticulum joins with the Golgi body, and in that way, during the protein movement, the proteins get modified, like there will be a carbohydrate that can be added or a lipid can be added in the protein. So these are the various modification that can be done in the protein depending upon the function of the protein in the cell. So with this I'm ending this class and I hope you have enjoyed the class. In the next class we will be talking about other cell organelles in detail. Thank you so much for listening.